Welcome, Brookings Bio students. This is Mrs. Rydell, and today we're going to talk about a second kind of cell division. And we learned about mitosis already, and what we're going to do is look at this new kind of cell division, see how it's different, see how cells use this kind of cell division to do something called making gametes. That's sperms and eggs, and it has to do with reproduction. So let's review a little bit about what we know about reproduction from our very first chapter. Remember, I said one of the characteristics of living things is that all living things reproduce. And we learned about two kinds of reproduction in our first chapter, asexual reproduction and sexual reproduction. In asexual reproduction, we're using the genetic material from one parent organism to produce an offspring. And in this case, this little flatworm does asexual reproduction. It's called a planaria. We have them in, they live in ponds and streams in South Dakota. You may see them when you're swimming or fishing. They look kind of like leeches, but they're not bloodsuckers. They're grazers. And these little worms, if you slice them in half, the head will grow a new tail, the tail will grow a new head, and you get two worms. The genetic material is coming from just that one parent worm. No sperm, no eggs. In sexual reproduction, both parents provide the genetic code for the baby. So dad makes the sperm, mom makes the eggs, and our offspring now is a combination, contains genetic material donated by both parents. And that's called sexual reproduction. And so let's look a little closer at those two kinds of reproduction. If you think about asexual, using the genetic code from one parent, when bacteria reproduce using binary fission, it really is a kind of asexual reproduction. The genetic code is coming from that parent bacteria, and copy the DNA, pinch in the middle, we're making another bacteria cell that has the same genetic code as our parent cell. Budding from plants is a kind of asexual reproduction. Some kinds of animals can do. We talked about that little flatworm can grow back a whole organism from the pieces. That's called regeneration. All of those are kinds of asexual reproduction because we're making offspring, making new organisms from the genetic code from just one parent organism. If you think about mitosis, right, mitosis is kind of like making clones, right? It produces cells that are identical copies of the parent cell. When we do asexual reproduction, there's some advantages to having an organism reproduce that way. First of all, you can make lots of offspring really fast. You don't need a partner around to do it. You don't have to hook up with someone. If you take those little flatworms and cut them into multiple pieces, one parent worm can make lots of, each of the pieces will grow back into a worm. So we can make lots of offspring really fast. Think about how long it takes to grow a human baby or an elephant baby, almost 2 years. So fast reproduction, and I don't need a partner. But asexual reproduction has some disadvantages too. Remember, since the genetic code is coming from just one parent, all organisms that come from that parent cell or that parent organism, they look the same. They have the same genetic code. And it makes it so that we don't have the ability for our species to change and adapt as the environment changes. One disease can come through, and because everyone's alike, all are susceptible. It could wipe out the whole population. Sexual reproduction, remember, we're taking the genetic material from two parents, and it makes it so that the offspring are different. They're different from the parents, and all the offspring that come from those parents are different from each other. Think about your brothers and sisters. None of them, unless you're a twin, none of them are exactly the same as you. The advantages of doing sexual reproduction is it allows for variation in the population. Everybody's different. And think about the people in this room. Look around. Everyone has different eye color, different hair color, different heights, different sizes, different shapes, different intelligences, different size feet. Everybody's different. And if you think about all the people on this planet, it provides a foundation. If a population is different, it provides a foundation for organisms as a group to change over time. That was one of our other vocab words from chapter 1. We're going to talk more about evolution and change in species over time later on in the second semester. But having variety in the population allows species to adapt and change. If a disease comes through, not everyone is going to be susceptible, and different organisms in a population are different, and that comes from sexual reproduction. So here's what we're going to do. We're going to take an egg and we're going to take a sperm when we do sexual reproduction, and we're going to produce an offspring, a baby. Mom donates the egg, dad donates the sperm. If you think about it now, if we made those sperm and egg cells the same way we did the other body cells when we talked about mitosis, making exact copies, if the sperm and egg had the same number of chromosomes, think 46 in humans, and I put a 46 chromosome egg together with a 46 chromosome sperm, I'm going to get a baby that's got 92 chromosomes. That is not a human baby. Extra chromosomes in humans cause some serious birth defects. And so what I want to do, what I need to do, is if my baby needs to have 46 chromosomes by joining an egg and a sperm, I need to figure out a way to make eggs and sperm that have half the number of chromosomes as a body cell. And that allows a 23 chromosome egg and a 23 chromosome sperm to make a 46 chromosome baby with the right number of chromosomes. So how do we do that? How do we make cells that have half the number of chromosomes? Because remember, mitosis makes cells that are exactly like the starting cell. So I'm going to take my starting cell, and we're going to do a double division and end up with 4 cells that have half the number of chromosomes. And meiosis is the kind of cell division that's going to allow us to do that. So we're going to look a little bit closer at meiosis. We're going to come back to this little video, and we need to talk about a little vocab before we look at the video. And so we need some vocab words here. Most cells, if you look at body cells, think skin cells, muscle cells, nerve cells, bone cells, they have 2 copies of each of the chromosomes. One came from mom, one came from dad, and we call those cells, if you look at those homologous, remember homologous chromosomes, they're called diploid cells. They have 2 of each kind of chromosome, 2 number 1s, 2 number 2s, 2 number 3s, and so on. And all body cells are what we call diploid cells. The short form of writing diploid, oftentimes we write it as call it a 2N cell, because there's 2 of everything, 2 of each kind of chromosome. All body cells are diploids, and the word for body is somatic. When we talk about somatic cells, soma means body. So body cells, think skin cells, muscle cells, nerve cells, liver cells, are all somatic cells. The other kind of cells you have in your body are what we call haploid cells. Haploids kind of sounds like half. Haploids have half the number of chromosomes as a body cell. They only have 1 copy of each of the chromosomes. We sometimes call them a 1N. 2N is diploid, 1N is haploid. And all sperm and egg cells are haploid cells. Remember, we want them to have half the number of chromosomes as a body cell. So when I put the sperm and egg back together again, I'm going to get a baby that's got the correct number of chromosomes. Okay, so let's compare a little bit just in general. Remember when we did mitosis, we're making 2 cells that are genetically identical. They're the same as the parent that we start with. They're the same as each other. And we're making what we call 2N or diploid cells. This is the way body cells make more body cells. And we're making somatic or body cells, used by organisms to grow bigger. That's how you got from a 1-celled zygote to a billion-celled multicellular organism that is 5 or 6 feet tall. We use it to repair injuries. Skin cells make more skin cells to fill in the hole or the injury. We use it to replace worn-out cells. And so when we're doing mitosis, we're making cells that are the same. When we do meiosis, though, we're going to make 4 cells. We're going to divide twice and end up with 4 daughter cells. But they're going to be genetically different. They're different from our parent starting cell. They're different from each other. And remember now, we want to cut our number in half. To make a sperm and egg, it needs to have half the number of chromosomes. So we're making haploids, or at one end cells. We call them gametes. Gametes are sperm and egg cells. And why are we doing this? We're making sperm and eggs so that we can do sexual reproduction. So what kind of body cells are going to be doing meiosis? Think about body cells. Where are you making sperm or eggs in your body? And that would be the reproductive organs. The testes, the ovaries are going to be doing meiosis, not a lung cell or a skin cell. Those organs can also do mitosis to make more ovary or more testes tissue, but they also do meiosis to make sperms and eggs. So what's going to make meiosis different now? Basically, there's three things, three kind of phases that are different in meiosis than mitosis. Number one happens in what we call prophase 1. Because we're going to divide twice and go through the cycle twice, the first time through we put a little Roman number 1 after the phases, and the second time through we're going to put a little Roman numeral 2 after the phases, so I can tell which division I'm talking about. So prophase 1, something called synapsis and crossing over, is different than mitosis. We didn't have that before. Number 2, segregation and independent assortment happens in anaphase 1. And number 3, we're going to skip interphase the second time. So no S, no copying our DNA. The cell is going to divide twice as it goes through meiosis, but it's only going to copy the DNA one time. So let's look a little closer at those three things. Remember, we talked about homologous partners. You have 2 of each kind of chromosome. And in your body cell, so in an ovary or a testis cell, each cell is going to have 2 copies of each of the chromosomes in my starting cell. And during prophase 1, those partners are going to find each other and form a little, kind of like a little group. They're going to stick together, and that's called synapsis. Synapsis. This little group of 4, now we have 4 chromatids. Now look at the arms. Remember, a chromosome is made up of 2 chromatid arms held together by a centromere. So I have one chromatid here, I have 1 chromatid here, I have 1 here, I have 4 chromatids together. That little group of 4 is called a tetrad. Tetra means 4. So we have a little group of 4 chromatids together, and that's those 2 homologous partners. They actually find each other, they stick together, they wrap around each other, and they make this little tetrad group. That's different. We didn't do that in mitosis. Once we match up our homologous partners, they can start trading DNA between the chromatid arms. And that is called crossing over, because a little piece of genetic material from, if this is my maternal, my mom chromosome that came from mom, and my paternal chromosome that came from dad, a little piece of the maternal chromosome, the mom chromosome, is going to trade places with a little piece of the dad chromosome. And notice the color change here now. Those pieces of DNA are actually switching position, and now we're going to have chromosomes that contain both a little piece of mom DNA and a little piece of dad DNA. It allows us to start mixing up, that's called shuffling, right? Shuffling the genetic material. We're mixing up the mom and dad gene choices in different combinations. And that's what's going to start to allow us to make sperm and eggs that are different from each other. Let's watch a little movie about crossing over and see how that actually works during prophase.
During prophase 1 of meiosis, homologous chromosomes come together as pairs. While lined up in this way, non-sister chromatids break at corresponding points. The chromatids then switch fragments, exchanging segments. This process is called crossing over. Crossing over allows chromosomes to exchange alleles. As a result, alleles are brought together in new combinations.
Okay, so let's go back to our slideshow and talk a little bit now about what is happening to our chromosomes. If you remember, homologous chromosomes are the same size and same shape, but they don't have to carry genes for the same traits. Dad could give you a green eye choice, mom could give you blue eye choices. They're not exactly identical. Remember, chromatid arms are exact copies, but homologous chromosomes are not the same. And so look at what's going to happen to my DNA now after crossing over. If you look at your mom chromosome and my dad chromosome, one came from mom, one came from dad, these chromatid arms are identical. They're copies of each other. This chromatid arm is exactly like this chromatid arm. If the little colors stand for different gene choices, for example, a green eye gene with a blue eye gene. Mom could give you green eyes, dad could give you blue eyes. Maybe on the other, farther down on the chromosome, we have some other traits like a red hair gene and a brown hair gene, right? And so mom and dad can give you different gene choices. And once those chromosomes match up and do synapsis at prophase 1 and they trade DNA, what we're doing is actually mixing up and putting different gene combinations together. This one is going to have green eyes and red hair. This chromatid arm that ends up in a different egg or a different sperm is going to have green eyes with brown hair. Blue eyes with red hair, blue eyes with brown hair. We're starting to make those different combinations. We're rearranging the DNA. So if you look at those homologous partners now, remember homologous partners were always different, but chromatid arms within a chromosome were alike. But now, after crossing over, these two chromatid arms aren't the same anymore. After crossing over, the chromatid arms are not identical anymore. And we're starting again to mix up the mom and dad chromosomes, those gene choices in different combinations. And that's how the same mom and dad can make kids that look very different from each other. Number 2, we said another way that meiosis is different is separation during anaphase, how the chromosomes go apart. Segregation and independent assortment. It's going to give us some more ways to mix up and shuffle the genetic material in different combinations. Okay, so let's watch a little movie about segregation and independent assortment. Another way to mix up the mom and dad chromosomes in different combinations.
The chromosomes of all organisms contain regions called genes. Each gene determines a particular trait of the organism. For example, one gene in pea plants determines seed color. Another gene determines seed shape. A diploid cell has 2 matching sets of chromosomes. Matching pairs of chromosomes are called homologous chromosomes. Homologous chromosomes carry the same genes in the same locations. Since all diploid cells have homologous chromosomes, each cell carries 2 copies of each gene. However, the 2 copies are not always identical. The 2 copies of the gene may specify 2 different versions of the same trait. Seed color, for example. One version of the gene on one chromosome may specify green seeds, while the version on the other chromosome may specify yellow seeds. The different versions of a gene are called alleles. The cell also carries 2 different alleles that determine seed shape. One allele for wrinkled seeds and the other allele for smooth seeds. Now consider what happens in meiosis. The chromosomes replicate. In metaphase 1 of meiosis, homologous pairs of chromosomes move to the middle of the cell. In anaphase 1, the homologous chromosomes are separated and pulled to opposite ends of the cell. 2 haploid daughter cells are formed. When the homologous chromosomes were separated, the alleles on those chromosomes were also separated. The allele for yellow seeds is now in the right daughter cell, while the allele for green seeds is in the left daughter cell. This process of allele separation is called segregation. The alleles that determine seed shape have also segregated. The allele for wrinkled seeds is in the left daughter cell, while the allele for smooth seeds is in the right daughter cell.
Okay, so let's stop here and we're going to go back to our PowerPoint and continue talking about ways meiosis is different than mitosis. if we talk about that idea of segregation now, and this is going to happen at anaphase 1, remember A for ana, A for apart or away, we're going to move our homologous partners apart during anaphase 1. And one partner is going to go to one daughter cell, and the other partner is going to go to the other daughter cell. That's the segregation. Think of another place where you have used or heard that word segregation before. Think social studies in history. At one time in America, we segregated our schools. We put all the white kids in one school, and all the non-white kids, the kids of color, in a different school. And that was called segregation. We're splitting them up, separating them. And so that term kind of applies to what the chromosomes are doing. We're splitting up the mom and maternal and paternal chromosomes. And one is going to go to one cell, the other is going to go to the other cell. And now if those go on to make a sperm cell or an egg cell, those sperms and eggs are going to have different gene choices. Remember, blue eyes, green eyes. Talked about seed color, yellow seeds, green seeds. When we split up the mom and dad chromosomes, because they are different, we're going to start making offspring daughter cells that are different, have different gene combinations. So segregation, independent assortment, we're going to do that crossing over thing. And then as we divide twice now, each of those chromatid arms is ultimately going to end up in a sperm or an egg, depending on if guys are doing it or girls are doing it. And look at the differences now. Because of crossing over, we're going to get different gene choices on the same chromosome. And because of that independent segregation of mom and dad chromosomes, we're going to start to see different combinations. The other thing that mixes up chromosomes is independent assortment. Remember, when the mom and dad chromosomes split into daughter cells, which chromosome goes with which is going to make a difference between what kind of sperms or eggs we're making. So if you have, think blue for boys, red for, we're going to do red for girls. These are the maternal chromosomes, chromosome 1 and chromosome 2 that came from mom. These are the chromosome 1 and chromosome 2 that came from dad. When this cell divides now, each daughter cell is going to get one of each kind of chromosome. It's going to get a number 1 chromosome and a number 2 chromosome. But which ones go together into our daughter cells, we have some possible different combinations. So think about the big number 1 chromosome from dad could go with the little chromosome also from dad, and you could get this combination. But the big blue chromosome could also go with the little red chromosome, and you can get some different combinations. That's the independent assortment. As they sort into the daughter cells, we're mixing up the mom and dad chromosomes in different combinations. Think about, look at how many different combinations we could get with just 2 pairs of chromosomes. Think about now 3 pairs of chromosomes. Here's my number 1 and number 1, one from dad, one from mom. Here's my number 2 chromosomes. Here's my number 3 chromosomes. And when I start mixing them up now, each daughter cell is going to get a big chromosome, a medium one, and a small one. But which of these go together in the different daughter cells makes a difference of what those sperm and eggs are going to look like. The big number 1 chromosome could go with medium from mom, big from dad, small from dad, or the big mom chromosome could go with medium and small from dad. And so we get lots of different combinations. It's the reason why, even though you have the same parents, you don't look exactly like your brothers and sisters. Everybody came up with a different combination of those mom and dad chromosomes when they were making sperms and eggs. So think about the possibilities of how many you could get if you had 46 chromosomes. The number is staggering, and that's the reason why everybody looks different on the planet. Okay, let's watch this little animation. This is showing us meiosis now. Remember, it's a double division. We're going to divide twice. And look at, we're going to match up our partners, crossing over, partners split, and then chromatid arms split, and we get 4 daughter cells. I'm starting with a cell that has 4 chromosomes. After the split, after the double division, I get 4 cells that each have only 2 chromosomes. 2 big ones, 2 small ones in our starting cell, a 2N cell. I'm going to end up with daughter cells that have 1 big and 1 small, haploid cells. And that's what meiosis does for us. We're getting multiple cells. We're going to get 4 daughter cells that have half the number of chromosomes. And the combinations of those chromosomes are mixed up. They're different. 4 daughter cells that are different. Okay, so meiosis, we're talking about making different cells. Crossing over, segregation, independent assortment, they're all ways of doing what we call genetic recombination. The daughter cells are different from the parent cell that we start with, and they're different from each other. So let's talk about phases now, and we're going to kind of walk through the phases and see actually what is different from the kind of division that you already know from mitosis. In mitosis, remember, we started with a body cell in G1 hanging out in the body doing its thing, and once the cell got the signal and decided it's going to divide, it would copy its DNA in S, it would make cell parts in G2, that's interphase, remember, and then we go into the cell division, prophase, metaphase, anaphase, telophase, cytokinesis. If this cell now wanted to divide again, it would go back to being a G1 cell. It could choose to copy its DNA and divide again, or it could take that detour out into G0 and kind of just hang out being a body cell until it got the signal again. In meiosis now, we're going to start in the same place. We're going to start with a G1 cell. And cell decides it's going to copy for whatever reason, gets the signal. S phase, we're going to copy the DNA. We're going to make the cell parts we need for the new cell in G2. That's interphase. And then it's going to go through the phases, prophase, metaphase, anaphase, telophase. You know that sequence, and you know what happens in those phases, right? And now if my cell now is going to do a second division, instead of going back into G1 like we did with mitosis, we're going to go back and start over again with prophase. We're going to skip interphase the second time. That's the third way that meiosis is different. Remember, we're going to skip interphase the second time through. So we're going to do prophase, metaphase, anaphase, telophase, cytokinesis again, without copying our DNA. We're going to divide twice, but we're only copying the DNA the first time through. So remember, this is a double division. So when I want to be able to tell which one I'm talking about, I put a little number. So prophase 1, I put a little Roman numeral 1 after the name. It tells me it's the first time we do prophase. Prophase 1, metaphase 1, anaphase 1, so on. And then the second time we go through, we call it prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2. We put a little Roman numeral 2 afterwards.